talk that's going to be given by Adam. Susan introduced you to Dr. Darwich's resume, and I just wanted to give a little bit of a background to his talk before Adam actually enlightens us with regard to what is happening in this particular space. Can we have the first slide, please? I thought it is important to actually to bring up a couple of items before Adam starts to tell us, what is going on with the model-based precision dosing, and that is one of them going and looking backward, what we used to do with regard to selection of the doses. This particular slide that you are seeing is trying to summarize what was happening classically, let's say half a century ago, when we were deciding on the dose. So, as a short summary, you can see that it is basically saying if 1 for man is adequate, then 2 for horse. And this summarizes the issue with the dose adjustments, which used to be only on the back of what we call allometry and size-based dosing. Obviously, things have changed since then. Allometry is used still with regard to the animal-to-human projection in many cases, but I think everyone now knows that size, even it is very important, it's not the only issue when it comes to personalizing the dosing. Next slide, please. So the question that you may ask yourself is that sometimes when we compare the pharmaceutics arena with all the other, let's say, parts and services that we have got, even if we take just buying a pair of shoes for yourself, why we are restricted to one size fit all is still in the pharmaceutical arena, maximum 2 or 3, let's say, dose bands, while when you go to a shoe shop, everybody knows their own size, they go and then they find, whatever that suits them. So one issue is that, what actually the measurement of the size and the necessity that we have got for the dose that is fulfilling cannot be obviously done by individual themselves. And the other one is, in this particular case, we have got multiple variables that they are coming to play. So we had a meeting recently to question why model-based precision dosing has been happening as a research, why it hasn't become popular, and Adam is going to hopefully to tell us more about the achievements, but at the same time some philosophical issues and hurdles that we have got on our way forward for that particular discipline. Next slide, please. So I think this is important for those people who haven't seen this document. This is a document that is now out as the PDUFA strategy for the FDA for 2018 to 2022, to realize that in the section that is dealing with modeling and simulation, that is page 30 and 31. This is a very long document, but I'm giving the pages, so if you wanted, you can go and explore. The modeling or reference to modeling in this particular case starts with actually PBPK. This is one of the things that we are going to hear more about during the talk by Adam. And then PBPK is aligned with several other modeling and simulations as a feeder. So it is not that PBPK versus others, but it is as part of the others, particularly establishing concentration effect relationship, which then on the back of PBPK enables us to define the necessary dose for each group, as well as the disease progression itself and so on. So with this background, I think hopefully we will get at the end of the talk by Dr. Darwich that why it is timely that we should be paying more attention to model-based precision dosing and how this integrates much of the information that we have gathered over the years with regard to the elements of dealing with the drugs, as well as many of the pieces of information that they are generated during drug development. With that, and without any further ado, I would like now to pass to Dr. Darwich to start his lecture. Thank you.
Thank you for the introduction, Amin, and thanks to the organizers. I can only concur with Amin that it's a very well-timed session to be talking about this. There's a lot of things going on in both research, health policy, and the regulatory space with regards to precision medicine. Just to give you a brief background, so the problem is really that there's a great lack of clinical data in special populations, and we see an unmet need for well-informed dose recommendations, both in the labeling but also in clinical care, clinical practice. So a relevant solution to this is, of course, model-based precision dosing and the PBPK that has showed really great potential in this area to address clinically relevant problems. So with the application of this precision dosing approach, we can expect an impact, and we're seeing it already actually on the drug labeling, dose guidance in clinical care and pharmaceutical development. So just to scale back the problem a bit, so it's perhaps best explained with this 3D plot that I borrowed from Amin. It's not a new concept though. So this one was first presented by Lou Shiner in 1991, and it describes the therapeutic response surface. So what you see here is that you have the patient characteristics against any dose-related factors like the dose, of course, DDIs, formulation properties, and that will determine the balance between efficacy and toxicity of drug. And really, this shape can look any different way, but the important thing is that when you take a single sample from this surface, for instance, you're not really describing the shape of the whole relationship. So traditionally, what tends to happen in clinical trials, you would take a very well-controlled sample from this surface and perhaps another one over here, let's say. And what you could do with that is using statistical methods like population PK/PD, you could infer something about the area lying in between these points, so you can interpolate what that dose-effect relationship would look like. The real issue is then when you go to more sort of patient-oriented populations or the real-world patients that will be taking these drugs, because they don't really sit within this surface. They might sit out here somewhere. So one solution for this is to use some form of mechanistic modeling. So if you use, for example, PBPK modeling that we're going to talk a bit about further on here. So the regulatory view now is that there are certain requirements in the label on issuing dose advice in relevant patient populations, and specifically the minimum criteria for that is pregnancy, women in labor and delivery, nursing mothers, children, and the elderly. So, of course, this is based on the totality of evidence, so it's not expected that we will have clinical data at day one in all of these populations, which I think is a sensible approach. But the real impact on this is the general lack of advice in terms of dosing. So I can just show you this graph here that I borrowed from the Framework on Dosing Recommendations in Special Populations paper. And it shows you the percentage in 2013, 2014 of approvals that did not give explicit dosing recommendations at initial approval for different degrees of renal impairment and hepatic impairment. So you can see for the severe degrees of both conditions, there is a great lack of any sort of guidance there. So they looked a bit closer on that data, and what they saw that only one of these recommendations, there was an attempt to use population PK analysis when the clinical data was lacking. There was mention of pregnancy, those recommendations for all the drugs, but in terms of real-life changes, they were rarely indicated. And for other special populations, there were really no useful recommendations in terms of dosing. On the other side of the coin, we see that there's an increase of the use of modeling to inform the labeling. So you see a lot of modeling carried out to predict metabolic DDIs for the label, and to some degree even special populations nowadays. And there's a lot of literature, mainly produced by academia, on model-based approaches applied to clinically relevant dosing problems. So really, a consequence of this general lack of guidance, you can see, for instance, in this paper here. So they looked at the dosing of vancomycin and gentamicin in 43 different neonatal units across the UK, and what they found that they could see 24 different combinations of dose or time of dose or time of monitoring across all of these units, and there were variations in how age was taken into account, and there were variation in how the banding was taken into account. So a lot of variability there in terms of what the clinical practice looked like. Same thing here, and perhaps an even more extreme example is this French study looking at antibiotic dosing in neonatal ICUs. And for 44 different antibiotics, they identified 444, that's not a typo, dosage recommendations for 41 antibiotics. So it's quite extreme and an extensive variation between the centers and how they tackle the dosing. So, I mean, the implication of this is, of course, the potential for sub-therapeutic treatment or even risk of toxicity in these patient groups. So the problem is pretty well defined, I think, by now. And partly the answer to that problem can lie in precision dosing or precision medicine. So we hear a lot of buzz around this phrase over the last couple of years. And really, it's not a new concept per se. So it's quite similar to what we previously called personalized medicine. But as I said, for various reasons, it's gained a lot of momentum lately. And as you can see here, so Obama says, what if figuring out the right dose of medicine was as simple as taking your temperature? So I interpret that as Obama being a quite strong proponent of model-based precision dosing. So some of the reasons why we see this sort of traction in the area is that there's a lot of new emerging, cheaper, more affordable technology. In genetic testing, we see new diagnostic tools, data handling, machine learning, for instance, and big data. And also you see in healthcare the move towards more centralized electronic systems to handle prescribing information. And really, the concept here, model-based precision dosing, sits within this precision medicine sort of discipline. And the idea is that We can use modeling in varying form of complexity to allow interpolation or extrapolation to special populations of dosage regimens, give the optimal drug treatments. So like Amin mentioned, previously this year, we hosted a meeting together with Certara on model-based precision dosing. And even though it was held in Manchester, many key opinion leaders in the field managed to show up. And we saw some great examples of the modeling being applied to healthcare questions. And we had a really useful discussion around why this has not yet become clinical reality. Although we see some pockets of very excellent examples in the area, we don't see this extensive policy of usage. There was also discussions around what the future potential could be of this approach to healthcare. So for the rest of the talk, I'll go a bit into the learnings from that meeting. So talking a bit about the general modeling framework, strategies for applying modeling to healthcare, and a bit about the future direction and implications. So based on our experience from the meeting, we can sort of divide the area of the model-based precision dosing into 3 different strategies that you could go down. So you have a real-time model implementation sitting directly in healthcare, the use of mechanistic modeling like PBPK models to do some form of extrapolation, or the perhaps more pragmatic approaches that you use a model at varying scales and you scale that down and simplify it into a dose-banding strategy that you can use in care. And around the framework for developing these models, of course, is quite similar to what we see for modeling and PBPK modeling for regulatory review. So one of the main drivers, of course, is the data that's available and the aims of the model. And for the more mechanistic model, you will more heavily rely on the systems data and the drug-specific data as well. So perhaps the key difference lies sort of in the validation stage, where we tend to see, first of all, the internal validation sets, then the usage of an external validation set at a different but related population, and then going on to clinical evaluation of the model, and at a varying degree of complexity, implementation of the model into a tool that can be used in healthcare. And of course, like you see for the regulatory reviews of the development scheme, this is a cycle where for each step you can go back and iterate on the performance of the model. So talking a bit more detail about these different avenues that you can go down. So for the real-time model implementation, you really see a lot of compartmental PBPK sort of modeling Bayesian analysis being used. And it's a very suitable approach for treatments where you see some form of therapeutic drug monitoring, so like immunosuppressants, etc., where you have a narrow therapeutic index and the risk of toxicity for sub-therapeutic levels. So the value of the modeling in this case will be really to either inform a more refined starting dose or reach target attainment faster compared to the more haphazard approach of adjusting the dose, taking TDM and then adjusting again. So a very neat example of this, so I took this from a paper by Michael Neely's group, in the Children's Hospital in Los Angeles. So it's looking at busulfan, an oncology drug that's routinely subject to TDM. And the drug is given IV, and there are also considerable variability there in exposure, and only around 60% of the patients will see target attainment. So during the dose adjustment, they typically take 3 to 9 samples to adjust the dose. So the idea here is to develop a model that can be used to perhaps minimize the number of samples needed and reach target attainment more quickly. So they developed a Bayesian compartmental model that they implemented in their local software, this BESTO software, and they managed to develop a strategy for dose titration based on only peak and trough sampling using optimal design applied to the model. And I think this is a very neat example in terms of the validation approach, because they worked with a lot of different data sets here, like you can see here. So they started out, of course, with their internal development set here of population, and then they used some in-house data to first validate it before using this larger validation set from a cohort in France. And finally, during the implementation stage, they did a clinical evaluation in-house as well. So that's a very neat example, I think, of this approach. Another really nice example here of the perhaps usability is from Cincinnati Children's Hospital. So these slides are courtesy of Sander Winks that works over there. And this is the interface that they have to support individualization in dosage regimens of immunosuppressants. So they have this web interface, as well as, if we look here, this software interface where you see information around drug monitoring data, so your TDM data, any patient pharmacogenetic data, inferred and measured adherence for the data that the clinicians can use to optimize the treatment. So, like I mentioned before, this type of approach is very suitable for TDM applicable areas, and this is also a good thing and a bad thing. Of course, the good thing is that you have the PK data there, but the little more discouraging thing is that you might not be able to produce a very large difference in your treatment or in your utility of this approach. So the difference in efficacy or treatment effects is usually quite minor. And one can argue that for some cases this requires some modeling expertise to be aligned with the healthcare, of course depending on what sort of user interface you're working with. It can potentially require fast turnaround, so you need to analyze your TDM data quickly, carry out the modeling, predict, adjust those, etc. So in other cases, you might not even be at the stage where you want to predict the individual patient's levels. You might be interested in knowing what's the average dose going to be for a particularly complicated patient population. So in this case, mechanistic models like PBPK allow extrapolation to do that. So from our own experience, we've been working on a bariatric surgery PBPK model. Really, the reasoning around this was that exposure pre to post surgery, although showing some relationship to solubility, is highly variable. So here we see the dose number function of solubility and dose against the fraction absorbed. and how highly variable the trend can be before to after surgery. So for some drugs, you see an increase in exposure, for others a reduction, and some of them are unaltered. So we did a service evaluation here at the local hospital in Salford, and just looking at the clinical pharmacy practices around these sort of cases. So what they tended to do was to go from a solid formulation over to either crushing the tablet or switching to a liquid formulation to potentially deal with the solubility issue that can occur after surgery. What really was a big guessing game as what was going to happen in terms of exposure. So our model that we developed performed well against different validation sets. So some of these data sets are, we have here observed and predicted exposure pre to post surgery. Some of these data sets are even prospective predictions that we did in order to carry out study design in collaboration with other academic groups. One of the examples, as you can see here, is cyclosporine. So that's an immunosuppressant. And this data is TDM data before surgery here, the first 3 dots. And then after surgery, together with data after surgery where they attempted to titrate the dose back up to pre-surgery baseline exposure. So you can see the gray area here are our predictions using our developed PBPK model. And we do a good job in capturing both the reduction we see in exposure, but also the titration back up to baseline exposure, you can say so. And the good thing about the PBPK approach, we can then go and say, what if we switch the formulation to neural formulation, which is the more solubilized formulation? We can compare what would exposure look like at the same dose levels as in this example up here. And we can see after surgery we have an increase in exposure, and that's due to this titration of the dose. So the exposure is not increasing in itself, it's just a dose titration that amounts to this increase. So further than that, we also had some, we acted sort of the clinical hotline with the local hospital here. So they would throw a few patient cases our ways when they weren't sure about what would happen after surgery, or if there was a particularly complicated case. Like in this instance, there was a male patient suffering from schizophrenia who was planned to undergo bariatric surgery. And of course, there was some concerns around his clozapine levels after surgery, as they wanted to maintain an efficacious treatment. So doing simulations here on clozapine, we could see that at the higher dose levels, there tended to be a reduction in exposure following surgery. And that prompted extra monitor in clinical care. And indeed, this was also confirmed in cases we have seen in the literature now for other antidepressants, where a lot of them tend to be poorly soluble, display a reduced exposure following surgery. The other example here was around a male patient who had undergone bariatric surgery and was on hydrocortisone replacement therapy, and he showed some signs of sub-therapeutic levels, and we could do some simulations to show that based on our best understanding, there wouldn't be a change in exposure before to after surgery. And again, in this case, they could confirm later on that hydrocortisone levels were maintained following surgery. So some of the difficulties around this approach is that doesn't necessarily cope well with high throughput. So if you don't have your models in place, it can be very difficult to answer these clinical questions in a timely manner that would make it useful. And of course, as you know, it's highly reliant on systems parameter data and drug-specific data. So the real question is here, can we simplify this approach somehow to gain more clinical utilization? The final approach and the more pragmatic way of dealing with this would be the sort of model-derived dose banding approach. So you develop a model and then you internally validate that and simplify it into a dose banding table with your significant covariates. So a really nice example of this one is pembrolizumab dose optimizations. So here in the UK, it's available in 40 mg vial sizes and is dosed based on kilogram of body weight. And because of this dose banding, you end up with a lot of wastage that has cost implications on NHS or the health organization. So the idea was here, can an alternative dose banding approach be used that takes into account both exposure and available vial sizes to maintain efficacy and minimize the wastage? So they could show here that using a modeling-derived approach, you could actually do that. So you could provide an optimal dosing strategy, taking body size and vial size into account, and resulting in large savings on the cost of the drug treatment. So it's a very convenient approach. There might end up being some impractical dosing intervals that could result in prescribing errors. You could also encounter the problem where you have multiple covariates that are influencing your dosing scheme. So how do you then go about to develop this sort of multi-dimensional dosing strategy? So some of the challenges that we identified to bringing model-based precision dosing and PBPK modeling and simulation into clinical practice. This is, of course, quite a multifaceted problem, but partly it's the lack of readily available clinical data. However, I must say, in the last few years, we are seeing a lot of initiatives where pharma are more willing to share data with researchers, like you see in Project DataSphere and clinicalstudydatarequest.com. And both pharma and FPR member companies have made some commitments to enhance the availability of their clinical trial data. Of course, another important step here is the availability of the systems information. So what will be your changes in enzyme and transporter levels in particular special disease populations? And this can be actually particularly difficult as in the case of obtaining samples from a population that is not necessarily very large. And of course, you all know the modeling approach requires a lot of drug-specific data. I think we are seeing a lot of things happening in terms of the generation of the systems data, especially now with the emergence of the proteomic analysis, for instance. Another factor here is that we see very little evidence of the utility or impact of using modeling in healthcare. And then I'm referring to actually a proven health benefit of using the approach, as well as a cost analysis to show that this strategy is optimal compared to the conventional approach. There are a few good examples from the literature, but in general, this area is needed of a lot of research. Hopefully, we will see some new initiatives coming out in that area. And I would say, as a community, as a modeling community, we have somehow we would need to see some improvements within our own groups, I would say. So we tend to pat ourselves on the back and say we've done a really good job with this modeling, but the modeling is just the means to an end and not the actual end in itself. So again, I would like to refer back to the clinical utility. And another question is whether we currently have the competence and the training of healthcare professionals to utilize these tools at the moment. And this also concerns attitudes to accept these sort of novel approaches in healthcare. And of course, we need to engage with the regulators as well as the healthcare providers to see some policy changes in the area. From the industry perspective, perhaps make little sense at the first glance to have a complicated, intricate dosing scheme. So the typical approach or the first intention is actually to avoid the dose adjustment. And for that reason, drug candidates are selected that have a wide therapeutic index and a low DDI potential in drug development. There are some benefits of this, so you have the simplicity of just using one dose, and you can say that it could be considered more safe as there are less risks of prescribing errors, for instance. You could also say that actually this sort of approach is hindering a lot of potentially beneficial drugs from being developed, as they are eliminated due to risks of DDI or a low therapeutic index, for instance. So a precision dosing strategy could help improve the safety and efficacy of these sort of drug candidates and bring them into markets. So I'll borrow these slides from David Fairman at GSK, and he just points out some prerequisites for what he calls the companion tool approach. So, for instance, in cases where you have drug candidates in a medical field that has a high unmet medical needs, you have the ability to somehow measure toxicity and efficacy, and there is a problem around the low therapeutic index of the drug. Then in that case, a viable approach could be this companion tool approach, where you at an early stage move forward to test a precision dosing tool together with your drug candidate in the early phase studies. And then what you end up with is a validated tool-drug combination that you use for treatments. So as I mentioned, this has the potential to be to bring some new efficacious drugs to the market that would otherwise perhaps be abandoned. And for other drug candidates, it can help maximize efficacy, minimize toxicity, and there could as well be a cost incentive in using this sort of scheme in your drug development stage. So around the negatives of this approach, you would of course require multiple dose strengths to be able to make a dose adjustment. Otherwise, that's not possible. There could be some increased potential here of misdosing, and there's the issue of who owns the precision dosing tool? Is it the pharmaceutical company, or is it another independent company, or is it healthcare itself? From the regulatory perspective, there's the potential here to improve the life cycle management of drugs. So you could, for example, see a scenario where you have an initial approval for basic dosing for a new drug, and then as further clinical data becomes available, you would see this sort of model-based precision dosing tool being integrated with the dosing strategy, and that could potentially be linked to some patent extensions, for instance. So there's no question about that we're going to see an increase in the use of PBPK modeling simulation to inform labeling. I thought I'd just bring up this graph here from a paper based on a meeting that was held in AAPS last year about model-based approaches to inform dose recommendation in special populations. So here we can see the information or available information against the needed dose recommendation. You can see here that kidney failure, hepatic impairment are highly attainable targets for your next drugs labeling. So no doubt we're going to see modeling being used to inform labeling in some of these areas, pediatrics as well, and obesity. So just to summarize again, so we do see an unmet need for model-based approaches to improve dosing strategies for special populations in healthcare. I hope I demonstrated some successful or potentially successful implementations that could be used. The responsibility is partly on us, the modelers, to provide healthcare professionals with the proven utility and cost benefits of some of these approaches. And there's really a good case here, I think, for pharmaceutical industry to adopt more of the modeling strategies to optimize dosing. And with that, I'd like to thank some of the involved parties in this. I referred to a lot of the material from other groups, so I'd like to thank them. And a lot of this material is as well generated during the healthcare summits that we held here in Manchester. Thank you for listening.